Good afternoon, everybody, and welcome to the Wednesday afternoon lecture. I am Preeti Chander, and on behalf of the NIH Fellows, I'm here to introduce our speaker today, Dr. Mario Amsel. Dr. Amsel is a world-renowned leader in structural enzymology. He's professor and director at the Department of Biophysics and Biophysical Chemistry at the Johns Hopkins University School of Medicine. Dr. Amsel's research focuses on understanding catalytic mechanisms of enzymes involved in redox reactions and phosphoryl transfers using various techniques, including X-ray diffraction, biochemical, and biophysical methods. His lab also does computational methods towards understanding thermodynamics of binding energetics. Dr. Amsel is not only a great scientist, but he's also a great teacher and an excellent mentor. He's won the Graduate Student Teacher of the Year Award and the University Teaching Award from alumni. In fact, after the talk, he's on his way out to receive a prestigious award by the Argentinian Ministry of Science and Technology, the RICES Award. Recently, his group described the crystal structure of PI3 kinase alpha, about which he'll be talking to us today. Thank you.
Okay, thank you very much, Preeti, for the introduction, and thank you, the WALS Committee, for the invitation to present my data here. My days have been delightful, incredibly good science, and very nice conversations with the postdocs. As Preeti said, there are several projects in the laboratory, and the one that I chose to talk today involves the structural determination and interpretation of the mechanism of activation of PI3K alpha. This is an enzyme which is not membrane bound, it's membrane associated. Here is the location, and is responsible to a series of receptors or their substrates, and is formed with two subunits. One large subunit, 110 kilodaltons, which contains the catalytic site, and one regulatory subunit of 85 kilodaltons that is probably associated with bringing the protein to the membrane. The project in the lab started when a group at Hopkins, directed by Bert Fogelstein, published in 2004 that the gene that codes for the 110 kilodaltons protein in this kinase was highly mutated in several cancers. Tumors have mutations in this protein, and that started our idea, say, why are these mutations involved? Since then, more data has been collected and more cancers happen to have these mutations. And by now there are about 1,500 mutations that have been described in the gene coding for the 110 kilodalton subunit of the protein. So most of the mutations tested increase PI3K activity. The ones that we don't know is because they were not tested. So this will be what people want to call a typical oncogene. Increased activity is associated with presence in tumors. The enzyme, surprisingly, is a lipid kinase. It has a very, very simple reaction. This is PIP2, which is phosphoinositide 4,5 bisphosphate, has two phosphates here. This is inositol. And what it does, it is phosphorylates position 3. That's all. And this reaction can be reversed by PTEN. PTEN is a phosphatase, which is a tumor suppressor factor. And that gives you the idea why they started looking at this gene in particular, because the reasoning was, if going in this direction, if you fail to have this reaction, you get a tumor suppressor factor here, then this could be an oncogene, and actually ended up being. So there are some questions that we can answer and some questions that we can, but some other people can, and this is why do so many tumors have mutations that increase PI3K activity? This is a very biological question and was sort of answered by other people. And then, how do these mutations increase PI3K activity is clearly a biophysical mechanistic question, and that's the general interest we had in the system. So the why is very simple. PI3K is here in this cascade. This is a membrane. These are receptor tyrosine kinases or growth factor receptors. When they become phosphorylated, the regulatory subunit P85 binds to them or to their phosphorylated substrates and bring this in an activated form to the membrane. This is the 110, and produce PIP3, the 3-phosphate, and that through a series of phosphorylation controls all these functions: growth, metabolism, survival, migration, motility, adhesion. Those are all characteristics that the tumor cell will try to develop, will develop through the number of replications that those cells have, a very, very large number before tumors become detectable. Just to give you a little bit more detail, there are two general classes of P110 chains. Alpha, beta, and delta belong to class 1A, and they use P85. And then P110 gamma has different regulatory subunits and also is not responsive to receptor tyrosine kinase receptors, but is responsive to G protein couple receptors, so it's responsive to chemokines. I'll give a little bit more detail about this. One thing which is interesting for people that are working in many of these signaling cascades, actually to get an idea of what are the proteins that actually control these processes, this is a complicated diagram and it's not complete, but that will give you an idea. This is where PI3K is, produces PIP3, that recruits to the membrane many of these, several proteins, plus AKT and PDK1, that then control through phosphorylation these proteins. Some of the actions are inhibitory, some of the actions are activating. But probably most of you recognize many of these proteins. These are the proteins that are involved in growth and translation. These proteins here are involved in apoptosis. So through the activation of some of these pathways, PI3K controls many of these activities. In a little bit more direct, all my talk is going to be centered around PI3K alpha, and just from the things that I show you, it's involved in several disease processes: tumorigenesis, because it's an alpha, is a human oncogene. In viral infections, some viruses require the function of the cellular PI3K. In cardiac function, they are involved in suppression of cardiac hypertrophy and ventricular dilation, fibrosis in situations under stress, and of course it's part of the insulin, the initial step after the receptor in the insulin pathway. So just as a general idea of what happens to the different classes, alpha, beta, and delta are class 1, gamma is class 1A, and gamma is class 1B. And these are the functions that they have. Just very, very simplified. I show you a little bit more detail in the previous slide. And these are the diseases that can be targeted by targeting these proteins. PI3K alpha is cancer because it has activating mutations, and in diabetes through the control of the insulin receptor. PI3K beta is involved in early development and platelet aggregation, and it's not mutated in cancers, but it has a function in cancers which are tumors which have deficient PTEN function. PTEN is the phosphatase. And then these two, although they belong to different subclasses, they are related with the immune system, and they are mostly involved in response to immune important cytokines that result in immune cell motility, and they are targets for inflammation and to immunity and asthma. So one would like to control somehow chemically these molecules for both, for biological exploration, for laboratory tests, but also for chemical intervention in disease. So there are several isoforms, and they are isoforms because the proteins are similar, that control cellular processes. For example, if one is thinking in cancer chemotherapy, one would like to inhibit only PI3K alpha because that's the only demonstrated oncogene of all these proteins. So in principle, the ideal compound should be isoform specific. But even inhibition, just blanket inhibition of PI3K will have some unwanted effects. So in the case of cancer chemotherapy, one would like to have compounds which are isoform specific but also mutant specific. So this is the general idea of where the realization of the individualized medicine at the level of the genotyping of individual of a patient will go. We are no, I mean, we can do it now, but for no use. The only use will appear when all the cell biology and all the biophysics is done and we know what to do once we know which genes are mutated. So the direction is to try to get all the information necessary to make genotyping of individual patients to be a clinical reality. So in the case of the design of isoform-specific and mutant-specific inhibitors, one thing that is crucial is biophysical and structural information. So can one actually design mutation-specific or very close isoform-specific inhibitors? And this is a cartoon, and shouldn't blame me too much for it, which is if you have a wild-type enzyme, you have an inhibitor. If you have a mutant, in this case is represented by having an additional cavity, it's clear that this inhibitor will bind to the mutant also. However, if you are able to design an inhibitor that fills that cavity, it will bind much less to the wild type. And this looks like a cartoon that it is, but in reality, we have seen that kind of differentiation in the case of one of the most studied for inhibitors enzyme, which is HIV protease. This is the wild type protease binding to one of the inhibitors, and then if you look at the mutant, this area, the interaction is completely different. So it's very clear that you could make, in this case you don't want to do that, but you can make an inhibitor that will be more specific for that mutant than it is for the wild type. So again, these differences are very difficult to realize by high throughput methods. They do require structural and biophysical information. So now let's look at a little bit of more detail on the structure of PI3K alpha. Is a 1068 amino acid, P110, the large subunit, is a 1068 amino acid protein, and it has 5 domains, an adaptor binding domain, a RAS binding domain, a C2 domain, a helical domain, and a kinase domain. The regulatory protein also has 5 domains, an SH3, a gap domain, an SH2, which is called NSH2, N-terminal SH2, and a C-terminal SH2 that are separated with a domain that unfortunately was called ISH2, but meaning intervening SH2 that is not an SH2, is a coil-coil. For the crystallographic studies, we tried over 50 or so different constructs, and the one that worked in insect cells was one in which we had the complete P110 and 2 domains of the P85, which are the NSH2 and the ISH2, and you will realize that this was not completely random. These are probably the most important domains to have present. We reported 3 structures, which is the wild type with the NI, with these 2. We reported one of the oncogenic mutants. I'm going to discuss this one, and the same oncogenic mutant in the presence of an inhibitor. And I'm going to just talk about the structure in general. In very few cases, we'll refer to something specific about these structures. So this is the overall structure of the protein, and here are the different domains. And it's a very complicated structure. The way the subunits appear has nothing to do with a linear sequence. And to review that a little bit better, I'll show you this. This is the last, the ABD domain. The RAS domain doesn't interact with this one. The C2 domain doesn't interact with either. The helical domain now interacts with these two, and the kinase domain interacts with the other three. So actually it's a quite complicated structure, and there are connecting pieces which are just there to connect this. Here is the coil, the ISH2, and there in the back was the NSH2. Just to get another general view, these are the subunits that I show you of the 110 kilodalton structure. This is the extended coil that runs all the bottom of the molecule, and this is the NSH2. I'm going to talk about these domains just in the sense what's their functional importance. So here we come back to the mutations that were detected, and you realize that there are two hot spots, one in the helical domain, one in the kinase domain. This is the domain that has the phosphoryl transfer activity, and there are some mutations in the C2 domain. And there are other mutations also that are found. I'm going to talk about these three mutations, started with the helical, then the C2 domain, and then the kinase domain. And one thing that probably will appear in one of my messages there, we look at the mutations not only just to see what the mutations do, they are activating the enzyme. And that is the physiological function, is to be activated upon interaction with receptor tyrosine kinases. So we expect that looking at the mutations is an easier way to looking at activation of the molecule in the physiological sense. So these are the questions. How do these mutations increase in activity, and how do these increases in activity relate to structural changes? So what we expect is what I just said. Information will be giving us insight about the physiological activation also. So we discussed many of these mutations in a series of publications, and there will be no time to write this down, but these are publications that probably discuss most of the mutations that were observed. And we are going just to discuss a few of them in some detail. So the first thing that happened here, the mutations that you saw occur here, this is the adaptor binding domain, this is the helical domain, this is the C2 domain with another set of mutations, and this is the kinase domain with another set of mutations here. So besides the mutations in the kinase domain, which is the catalytically active domain, all other mutations occur at domain interfaces. So one thing which is also important, I had to turn the molecule 180 degrees to show you where the binding site is. The catalytic site is here, which is here. All the mutations are very far from the catalytic site, yet they are all activating. So how do they do it? And for some of the mutations, the answer lies on this subunit here, which is the NSH2 domain of the regulatory subunit. So it was known from biochemical data that the NSH2 domain has a partial inhibitory effect. The story is complicated, but if you look at the details, you need the regulatory domain for the stability of the protein. But with the regulatory domain, the protein is less active than without it. So, and it was determined that that was due to the NSH2 domain. So this is slightly inhibitory. So one way that some mutations may work is by relieving the inhibition by the NSH2 domain. So now we are looking into the structure straight into the NSH2 domain. Here is a more realistic show just of a surface of the NSH2 domain, and you realize that it spans a place where there are 3 domains of the large catalytic subunit present, the helical domain, the C2 domain, and the kinase domain. This is where the binding site is. So here is where the NSH2 domain is present. It's far from the binding site, but it seems to interact with 3 of the subunits at a very, very crucial point where they just control the position of each other. One of the strong interactions occur between the helix of the NSH2 domain and the position of the helical domain and the kinase domain. So in this group, this helix is there. That's one of the important interactions. And we are going to look at this interaction from 2 perspectives. One is from the perspective of the binding of phosphotyrosine peptide, which will be the equivalent of binding receptor tyrosine kinases or their substrate. They are just phosphotyrosine signaling groups. And the other are these mutations that are frequently mutated in cancer, are those related to the physiological activation. So we were not able to get the structure of the whole protein with the peptide present, but over 10 years ago, the structure of this identical domain, isolated from the protein, in the presence of phosphotyrosine peptide, was determined. I think that it was determined twice. I think this is the one from the group of Steve Harrison. And this is the NSH2 of the same molecule with a phosphotyrosine peptide bound here. If we align this NSH2 with the one from our structure, they are identical. So in principle, by this trivial modeling, we know the position of the phosphotyrosine peptide in our structure. So this is where it would bind. This is the NSH2 domain, and this is where the phosphopeptide model phosphotyrosine peptide will bind. If we now look at the complete structure, that same groove is occupied by a loop, and that loop carries the 2 hotspot mutations of the helical domain, glutamate 542 and glutamate 545. So essentially, whatever the phosphotyrosine peptide is doing, something very similar is being done by these mutations. They just are in the same place. So after the structure was published, a group of Luo and Carson published the following enzymatic data on the wild type and some of the mutants. And here is the activity expressed as a Kcat over Km. This is the activity of the wild type. This is the activity of one of the helical domain mutants. This is the activity of the other. And here are the data. The increase in activity, the activation is always in signaling molecules of the order of 2 or 3. Very, very small activation, but is important for initiating signaling cascades. If we now normalize these three activities to 1, and those are the white bars, then this is the activity without anything added. If we look at the black bars, is how much the activity increase if you add phosphotyrosine peptides. And the wild type gets an increase in activity by about a factor of 2, which is the correct activation. The 2 mutants that I described do not increase their activity, which means they used up already the same activation mechanism, which is removing the NSH2 from its inhibitory position. So this is more or less what we think happens, and that is the NSH2 domain exerts an inhibitory effect on the kinase domain, and that inhibition can be relieved by binding phosphotyrosine peptide, which will move this somewhere else, the NSH2 domain, or by having these mutations in the same surface that weaken the interaction of the helical domain with the NSH2. So the things will look more or less like that. This is the molecule, and we are looking now at mutations here. And this group, this domain, is inhibitory of the overall activity. So here the inhibition is shown by this arrow. If we have a mutation here, or we bind peptide, we dislodge the NSH2 subunit, and that results in activation. So that will be the general scheme for the mechanism of activation by a receptor tyrosine kinases or by mutations. So there is a catch to all this. In the mutations, the idea is simple, but what happens when we try to look at how will the physiological activation occur? For the physiological activation to occur, this space has to be vacated. Otherwise, you don't bump things out of the protein. There is no enough binding free energy close by to be allium starts to bind and then moves and pushes somebody away. That doesn't happen. This site has to be unoccupied part of the time. So that means that there is some sort of dynamic equilibrium between the helical domain and the NSH2 being bound to each other, and some part of the time the NSH2 domain is accessible to bind phosphotyrosine peptide. So one has to look at dynamic effects. And to look at dynamic effects, we went in two different ways. The protein is too large and we don't know enough NMR, so we try to do it in the ways we know. One way is to just doing computationally and try to see what kind of conformations are accessible to the molecule. Again, because the molecule is very large and one wants to have indications of large motions, we went to do normal mode analysis. This is one of the modes, and I'll show you some more, exaggerated a lot just for visualization purposes. So we did normal mode analysis in the wild type and in the mutants, double mutants, in the positions of the mutants of the helical domains, which are the ones that I discussed. So the first thing that happened is that when you try to model that mutant, the arginine residues instead of the glutamates, for example, just push the structure away, and when you look as it compared with the wild type, the NSH2 domain is already slightly moved away. And what we try to analyze is what's the effect of weakening this interaction, it doesn't disappear, on the general dynamic motions of the molecule. So what we were describing as the dislodging of the NSH2 binding to the large domain is more than anything is weakening the binding, and that gives access to dynamic states which are away from the position it takes normally. So one thing we looked at in detail were the dynamics of what can the NSH2 domain do, and here are a large number of normal modes. What it is represented is in arbitrary units, the amplitude of movement of the individual residues of the NSH2 domain in different normal modes. And here in the movies they are being represented the one of the first normal mode. And if you look at this, what it says is the wild type you never see it because it's the gray line. It has a very, very small amplitude of motions for the NSH2. Once you weaken the interaction of the NSH2 by the mutations, you get very large fluctuations that are possible at low energy cost. So here is the NSH2 of the wild type, and here is the NSH2 of the mutant, and there are much larger amplitude motions in the mutant than they are in wild type. And those motions changing the interaction of this region with respect to the kinase domain, which is the purple domain. So one can look also at just a very, very general idea of what the motions are. And here are the average over two groups of normal modes. And in orange is the wild type. And these are, again, general amplitudes of motions as a function of amino acid number. In green is one of the double mutants. In gray is the other double mutants. And it's very clear that for the NSH2 domain, the amplitude in every respect of motion becomes much, much larger than that of the wild type. And in general, this is the interaction that is weakened, and that is where the peptide is going to go. In this case, these large movements are the ones that directly activate the enzyme. These large movements in the wild type, in the orange, are the ones that will allow the receptor tyrosine kinase to bind. The ISH2, the long helical domain, for example, doesn't experience the same kind of motions. Native, there are motions, but native and mutants are extremely similar. You cannot recognize here, you cannot recognize here which one is green, which one is orange, and which one is gray. The same is true. There are two loops in the molecule which are important for activity. For the moment, I said we dislodge the inhibition and we gain activity, but there should be a mechanism of gaining activity. And that part, of course, we don't know yet. That's what we are trying to do. But one thing that is clear is there are two loops that are called the activation loop and the catalytic loop that have very, very varying conformations when you look at a very large number of kinases. And here we build a model of the activation loop, and then we look at its fluctuations, and it's the same color code. The wild type is orange and the mutant is in gray. And there are many movements that are large amplitude for the mutant, which are not large amplitude for the wild type. We expect, when we analyze them, that some of these movements will show some positioning of residues that increase catalytic activity. We are not there yet. Here you can see we are representing again the one with the red dot, and there are some large movements in the wild type, but here where the loops are, the changes are very small. If you look here in the activation loop, the changes are very large. So again, the dynamic effects will make conformations accessible part of the time, conformations which are more active catalytically. They are not static conformations, they are just become accessible. There are many structures that have been done by us and by others of this, and there doesn't seem to be a hemoglobin type structure where you have one structure here and one other structure here. There are changes that occur locally and changes in the dynamic like this one, which seem to control the catalytic activity. Remember that the whole increase in activity is only a factor at the most of 4. Okay, so there are very, very subtle changes in activity. So just to give an idea, what we said here is that the mutations weaken the interactions, changing the characteristics of the states which are accessible dynamically. And then phosphotyrosine peptides and the mutants have similar effects. So one thing that we decided to do is to try to see if we can get any information about binding of peptides to the molecule. The molecule, probably because the NSH2 is dislodged and it is just moving, is resisting to be crystallized in the presence of peptide. So we went to a technique called small angle X-ray scattering. So in small angle X-ray scattering, the kind of information you get is an envelope for the molecule. The envelope here is shown as this egg crate, just because of the method that is used to calculate that envelope. And here in this envelope is the fitting of the molecule. The fitting looks much smaller than the envelope itself, but these envelopes includes water which is immobilized, or at least is tracking the surface of the protein. So probably these things reflect water associated with the molecule. When we add phosphotyrosine peptide, for example, from the insulin receptor 1 that we know bind to the NSH2, which is here, this portion of the envelope changes dramatically. There are other places where it changes, and we have some explanation for this, but I'll concentrate in this portion. This is where the NSH2 is, and this is the new portion of the envelope. So what we think is happening is that this NSH2 now can sample all the conformations between these 2 points, and now the envelope becomes bigger here, so NSH2 samples from here to here. That is consistent and similar to the observations that we had from the normal mode analysis. The data, the experimental data here, and you realize the information content of this data is very small. This is all what you get, but it has enough information. These envelopes are then completely unbiased. They do not reflect anything of the structure. They are then just based on the experimental data. And then we did it with another peptide just to make sure that that was not a fluke. And we did it with several peptides. Here are all the peptides. These are all the experimental curves. Here is the envelope with another peptide from another receptor tyrosine kinase associated protein. And again, this region has grown with respect to the wild type. Each of these envelopes was calculated 10 times with 10 different data sets. So there is a real signal there. So that's more or less where we are. We are now trying to look with more sophisticated ways of matching the experimental data that take a bigger advantage of knowing the possible coordinates of the structure or knowing the dynamics, just adding all the conformations that are being sampled in solution. Just to summarize this part, the binding of this phosphotyrosine peptide releases the inhibition by weakening the interaction with the NSH2. Dislodging the NSH2 from the central position changes the states that are dynamically accessible. It's not an observable new conformation. It's just conformation in which something becomes just spends more time away. These changes, once these happen, they are changes that are in the conformations accessible to the catalytic loop of the kinase domain that probably will be able, or somebody will be able to find that they will have changes in the catalytic activity. There will be some of these conformations that have the correct organization of catalytic residues. And the effects of phosphotyrosine binding seems to be the same as those by the oncogenic mutations in the helical domain hotspot. Another group of mutations occur in the interaction of the C2 domain with the helical domain. This is a surface representation of that big rod, the coil, and I don't have time to discuss it, but there are a series of specific interactions which involve charges and hydrogen bonds. But there is a large surface of interaction. There is one mutation which is quite prevalent in the C2 domain, and this is asparagine 345. Asparagine 345 in the structure makes hydrogen bonds to two residues of the coil of the ISH2 domain, which are asparagine 564 and aspartate 560. So we reason that changing this residue will change these two amino acids and will have some effect on the activity of the enzyme. That seems to be true, but there was another prediction that one could make is, why did nobody found mutations in these two residues? They will have the same effect. The cancer cell will be as happy with these two mutations as it is with this mutation. And the reason is that they didn't look first. So about 2 years after the structure, the sequence came up of a large number of glioblastomas. And when they did that, that's from their paper, they found that the residues that were predicted that will have the same effect are mutated very frequently in glioblastomas. So it's very clear that we are getting a handle on what these mutations are doing. So in the case of the mutation, if we use the same cartoon diagram, this mutation is here. And now we'll dislodge this. So the general idea is this is the inhibitory interaction. If we have a mutation here, this is a rigid rod, is a coil. So just changing the position of by a mutation here of this rod will also have an effect on how strong is the interaction of the NSH2 with the helical and the kinase domain. So mutations here will have the same effect, which is weakening the interaction of the NSH2. So to just discuss the last kind of mechanisms, I'm going to talk a little bit about membrane interaction. This is a lipid kinase, so the substrates, which are phosphoinositides, are membrane components. They are never in solution, they are membrane components always. So the protein has to somehow interact with the membrane. Some of the things are easier than others. If you look at this surface of the molecule, it has a higher percentage than usual of blue is positively charged, of positively charged residues. Here are their names, but it's a very large number of lysines and arginine residues. So we another propose that this surface interacts with the membrane. I'm going to skip that. So this is what the membrane would be, and it's interesting. This is the ISH2 domain that in reality had not been predicted besides a paper, a very old paper using antibodies that will interact with the membrane. This is the adaptor binding domain, but also the C2 domain interacts very strongly with the ISH2, and the C2 domain is a membrane interacting domain, so everything makes sense that this will be the general surface. And the catalytic site is somewhere here, and I'll show it to you in more detail, and the access to substrates. This is another representation of the same, and here is where the catalytic site is. I cannot see it from here, but maybe the screen is brighter in your direction. There is an ATP there built in the, for the ATP site of the catalytic site, and here is a direct connection to the membrane, where you realize this could have been a little bit more in the membrane, and then the head groups will be accessible directly for phosphorylation. One thing that is interesting, these are one set of interactions which are directly non-specific interactions with the membrane by domains of the protein. However, there is a structure of another isoform with RAS. So RAS binds to the RAS binding domain. We transfer that binding to our model, and then this is the C-terminus end of RAS, and this is more or less what the length of the part that was not present in the structure because of disorder, and that's where the anchor, lipid anchor for RAS is. So it's possible that at some conditions, the function of RAS is to aid in anchoring the protein to the membrane. And there are some stories that you can think related to that. So again, this is where the binding site is, and this is where the phosphoinositoid lipid substrates will be. So what happens with this mutation, and why am I mentioning the membrane when I talk about that mutation? This is one of the most prevalent mutation, and it actually correlates quite strongly with aggressiveness of the tumors and survival. It's located here very close to the end of the molecule. The molecule has 1068 amino acids. This is amino acid 1047. It's usually a histidine. It's most of the time an arginine in the mutant. We did the structure of the mutant, and there are some small differences, but I showed you the structure is almost identical to the one of the wild type. In just general, all the subunits are in the right place. So what is different? So there is, if we look at the part where residue 1047 is, here is what 1047 is in the wild type. The wild type is a histidine. The histidine points in that direction, and then the cell, the chain is completely disordered after one amino acid after 1047. We never saw any more density. When we look at the mutant, many things happen. Is this site, this whole conformation changes because the arginine now points in that direction where it can interact. And now this whole loop up to amino acid 1062 becomes ordered. For this loop to be ordered, this other loop has to change conformations, and these two loops now form a surface here. But if you look at our suggestion of where the membrane portion is, we change completely one of the regions where the protein interacts with the membrane. And here are the two situations. This is the wild type, and this is the mutant with the two loops interacting with the membrane. We did some calculations, and the calculations indicate that it's not only that, but there is some change in orientation, a small change in orientation with respect to the membrane. This one will move in that direction to make a bigger contact. And more or less, it says that we are increasing the interaction energy with respect to the membrane by about 1.2 kilocalories, which is about a factor of 6 to 8. So seems to be that this mutant interacts a little bit better with the membrane. If that is the case, that means that it may be changing the activity by just having more access to substrate. If that is the case, this mutant should be activatable by the physiological activator of a phosphotyrosine peptide. And those experiments were done by Luo and Carson, and this is the same slide I showed you before. The wild type here is the rate just as a Vmax. The wild type has lower activity than the three mutants. These are the ones that I discussed before, and this is the new mutant, more or less the same Vmax. If we now normalize them, those are the white bars, to 1, and then we test them in the presence of activating peptide, the wild type increases, as it should. These two don't because they use the same mechanism, but this one is further activated by peptide, which means whatever makes this activation is not release of the NSH2 activation, inhibition. So clearly there are two mechanisms at play. One is relief of NSH2 activation, the other is increase of the accessibility of substrates. Just, we are not a great membrane chemists, so we had to devise an experiment that even we could do. And in what we did, we said is, if this is operational, just differential access to the membrane, we should detect it somehow. So we did the assay in the presence of just vesicles that contain different phospholipids, phosphatidylserine, liver and brain lipids, and lipids from two colon cancer cells, DLD1 and HCT116. They were used, these cells, because they were available in Bert Fogelstein's laboratory, not because we want to put any emphasis on the fact that we are tumor cells. And we checked for the incorporation of a P32 from ATP into PIP3. And when you do that, if you look at the wild type and the mutant, here they were normalized to 1 for basal activity. If we check in phosphatidylserine, if we check the different lipids, they respond to the change in lipids in very different ways. So not very clear that everything that we said is proven, but what is proven is that the mutant with these changed loops has different way of interacting with the membrane than the wild type. Okay, one thing just before I finish, and I'll have to skip some slides, what happened, what do we know about binding of inhibitors? So the one that we tested was Wurth-Manning. Wurth-Manning is a general kinase of this type inhibitor, and what it does is it makes a covalent bond. It does bind in the ATP site, but has a covalent bond to a lysine residue. This is the structure, and that is where it is. That's the way we know where the ATP site is where Wurth-Manning binds. In this drawing, this is where the membrane is, so you see how the access of the membrane to the catalytic site is. Oops. So if we look now at two isoforms, and let's look for isoform-specific inhibitors, this is the isoform gamma, which is as far in the isoforms of alpha as you can get. If you look at the binding of Wurth-Manning, this structure was done by the group of Roger Williams, and this is our group. All the residues, K, K, S, S, I, V, V, all the residues which are involved in interaction with Wurth-Manning are the same, are conserved. So it's very clear that why Wurth-Manning is not a specific, isoform-specific inhibitor. It's a very general inhibitor. However, when we looked at the structures in more detail, we saw that this loop in the alpha experienced between unbound and bound a large change, while this loop here doesn't. In the case of gamma, it's the other way around. This loop doesn't experience any change. This one experiences a very large change. So even if the structures are extremely similar in the binding site, it would be possible to do isoform-specific inhibitors by looking at the conformational, local conformational effects of inhibitor binding. I have more things like that, but I think I'm going to skip them. And just to give you an idea what other thoughts occur when you look at the binding sites. This is the ATP binding site. It's located here, and I show it to you the relation with the membrane. This is a very well-studied site, and all the inhibitors up to date are inhibitors that go to the binding site because they come from the background chemistry of kinases, and that's what they have in common, not the substrate, but the ATP. It's very difficult to find a phosphinosytide binding to these proteins. It's very difficult to see it in structures. So we look for it. We have some experience in looking for phosphinosytide sites in structures. And we came up with this site is in the correct position to be phosphorylated. This is the 3 position that will be phosphorylated. The relation with the membrane is perfect. So we are now exploring this site as the possible phosphinosytide site. That will give the idea, open a new site for the developing of inhibitors, are also for bisubstrate inhibitors, just inhibitors that cover the ATP and the phosphinosytide sites. And that's something that we are exploring just from the biophysical point of view. So just to get an idea of what I tried to tell you in these 50 minutes, there seem to be 2 general mechanisms of activation. One, which is the release of NSH2, and that's clearly related to receptor tyrosine kinase binding and some of the oncogenic mutants, and then change of the interaction with this enzyme with the cell membrane, and both of them are present in mutations, but both of them are probably important physiologically. So how is the relief of activation? The NSH2 domain is a scaffold that contacts 3 domains and possibly just modifies the communication between those 3 domains. Activating phosphotyrosine peptides relieve the inhibition by binding to the NSH2 domain, which is the domain that is modifying the interaction. However, access to this site by the receptor tyrosine kinase requires that this domain is at least part of the time removed from this position. So there are clearly a need for dynamic effects. For the mutations, many of the mutations occur at interfaces between domains, and some of these mutations use the same mechanism as the physiological activation, and the mutations can occur either directly in the interface or they can occur very far, and when they occur very far, they are communicated by this coil, which is a rigid rod part of the molecule. The effect of weakening this interaction is a change in the states that are dynamically accessible. They are probably not very well-defined conformational changes. And in the case of inhibitor binding, it's possible that isoform-specific reagents can be made by looking at the conformational changes that occur after binding the known inhibitors. And then the other mechanisms involves interactions with the cell membrane and interactions with the cell membrane. Sensitivity of the interaction with the cell membrane correlates well with the structure and the activation by this particular mutant. So I want to thank the people. You probably see the name. Sandra Gabelli worked in this project from the beginning. She directed most of the day-to-day operations of this work. Bear was a student that did the structure of the wild type. Krishna works on several aspects of the project, including expression. Evan Brower is a postdoc that worked on some of the expression of P85 and things I didn't talk about. Ignacia Echeverria is a graduate student that is graduating in a few weeks, and she did all the calculations that I showed you, and she directed her SAM that was a rotation students. In the laboratory of Bert Fogelstein, Diana did the structure of the mutant and the Workman structure. Oleg Schmidt did all the reactions, the biochemistry, and the other are collaborators from the laboratory of Bert Fogelstein. And we did many of these experiments in Brookhaven National Laboratory, funding of the NCI and the NIH to Bert Fogelstein, and we started this project on another phosphoryl transfer project and evolved into this project, and for that other project we had funding from NICMS. And I'll show you the people in my lab. This is a very not light photograph, but here are some of the people I mentioned. Here is Ignacia, who did the calculation. She's Krishna, which was involved in expression of several of the proteins. That's Evan. And am I missing? Oh, here is Sandra, of course. And I thank you very much for your attention. So I'll be glad to answer questions. I'm sure that not everybody believed everything I said.
Before people leave, there's a reception at the library after this.
Yes.
Use the mic, please.
Oh. Who has a mic? Oh, the mic there. Oh, wow. Okay.
Are the mutants who works on different domains but supposed to work on the same mechanisms, are the activations of the kinase is synergistics, or it's they work completely, I mean, you get the same effect?
In that case, it was when tumors are sequenced, they usually you don't know if the mutations occur more than one in the same cell. They are just conglomerates of cells.
But
so we did not try mutants, double mutants that were separated in sequence. We tried this hotspot or this hotspot. And as far as I can tell, double mutations of distant mutants were not checked to see if they were synergistic.
But to get the drugs that actually will work, will be mutant specific, you would like to have mutant that works on different domain but works by the same mechanism, not to be additive, obviously.
That is correct, yes, yes. The only non-additivity was shown with respect to phosphotyrosine binding, but not to mutants, correct. And that will be a good experiment, yes. Yes.
So you talked about dynamics a lot. What do you think, on which time scale does the protein behave dynamic? I mean, do you think it's fast or it's rather slow?
You realize some of the calculations were done at 3 levels. One was dynamic, elastic dynamic networks, anisotropic networks, and then full atoms. So all the frequencies that we are looking at are, let's say, low frequencies, but the time scale is difficult to believe. But this will be, you know, the frequencies as you get them are of the order of 1 centimeter minus 1, so then they will be of the order of 10 to the 7 per second or something like that, which is, I think, difficult to believe. So the time scale is difficult to estimate. It's possible that, you know, it does reflect reality. It's possible that it doesn't. It does reflect the movement. For those to occur at a stable structure, it has to be easy to move in those directions. That's what these calculations reflect. If you are at a stable structure, those directions that we are seeing are the ones where it's easier to move. Once you move from the stable structure, how far or how fast is much more difficult to decide. But in a system of this size, unless, you know, new techniques for NMR become accessible, it's very difficult to get timescales, yes.
Sorry, just one. You
Yes.
said the activity was not that much higher in the mutated proteins, right?
Sorry, say it again?
The activity was not that much higher, so it was not something like tenfold or so. It was.
No. That was the biggest surprise to me when I got into this. You know, I got into, I come from other phosphoryl transfer enzymes, and, you know, we do a mutation. A student comes back to you and says, you know, we made this mutation, is a factor of 3, and you say, well, that has nothing to do with the activity, with the mechanism of the enzyme. That would have been my standard response. Then I get into signaling, and very small changes have very large effects. I'm not completely sure how that is accomplished, in the sense that if you actually do have that basal activity, why are things not activated all the time? Clearly depend on many other situations in the cell. You could say that you have a very low basal activity, but P10, which is the phosphatase, it's always just taking it away. But something very silly to do. Cells are known to do silly things, but why would you phosphorylate something and then do a kinase just to keep it in control? Do a phosphatase to keep it in control. So I'm as puzzled as you are. I shouldn't be, but I am, yes.
Any more questions? If not, let's thank the speaker, and please join us at the reception.